So I think, yes, today I had as the deadline for project proposals, and I didn't record it last time, so Christine had some questions because I typed it up and we talked about it and then I started recording. So remember, it's just about anything, but on our modules, you know, I suggested some chapters in your book you could look at, but pretty much anything genetic related you can propose to me by today. I will definitely try to get back to you guys by Thursday, right, because projects are due November 8th. You have the option for bonus points of presenting at the STEAM Day, right, which is November 14th, which is a Monday. It runs from 10:30 to 3, actually really starts at 11, right? Setup is from 10:30 and ends at like 2:30. Just if you are a working person and you want to try and ask off for that day, that's why I gave you guys times. You don't have to be there the whole day. As long as you put in at least an hour, you'll get your 10 bonus points if you show up and participate. But keep in mind the audience is 5th and 6th graders, right? So, you know, if you want to do something sex related, I would suggest not. Right? I don't know how the teachers will feel about that. I'm in the process of securing the schools that will be coming and at what times they'll be coming. So hopefully that'll be put together soon. But I already have the location reserved. It's the 2nd floor in the Student Life building, the whole big Pontchartrain Center. I managed to get it reserved. My boss still doesn't know how I managed to do that, but I'm good like that. He's kind of jealous. He's like, every time we try to reserve, they're always booked with Goldman Sachs. I said, well, I worked around their schedule too. I went there and literally said, okay, what day do you have available that I could use, and figured out which ones I could do. So it took some finagling. I'm open to any type of format, but please let me know, right? Because you need to know how you're going to be putting it together. You know, you could do a PowerPoint. Obviously, if you're going to present at the expo, you might want to do a PowerPoint and then stick it on a poster board, or just do a poster board. You'll also have the option, depending in this class, if we have time towards the end of the semester, you could present to the class for bonus points as well. But you don't actually have to present it to anybody. You can just hand it in, your poster, your PowerPoint, any other format that you want to do. If you're one of those fancy people and you want to do a video, right? I'd take that, right? Anything. I'm really open to anything you want to do. Just you got to propose it to me so I can look over it, give you suggestions, guidelines, and such as that. Okay? So get that done today if you can.
How detailed do you want the topics?
So the more detailed, the better. But it can be, you know, literally like a disease. Right. So also I just want to make sure, not that we have that big of a class, that we don't have any repetitive stuff either. But also to be able to kind of guide you guys, right, too. The first part is picking, and then you start researching, right? And along the way, I'll be asking you guys to check in with me. Make sure you're on the right path. But it's a genetics class, right? So if you're doing a disease, it's got to be a genetic-based disease, right? And you got to talk about the genetics involved in it, okay? But I'm really open to format and presentation format and all that, okay? All right. So for submitting it, I'm not in it, but it should give you a little box. You hit submit. I suggest typing it up and just pasting it and copying it into that box. Don't type in that field, because if you lose an internet connection, you lose everything you wrote, right? So it's best just to type it up and then just submit it to me, right? Paste and copy from like a Word document. Okay, so we're on Chapter 9, and I skipped over the Hershey experiment because I wanted you guys to go over that on your own, the Hershey Chase experiment. And I apologize because I actually had posted this PowerPoint before class on Thursday, but I didn't turn the link on for you guys. I didn't publish it. When I came back later, I was like, oh, well, nobody complained, but I actually had it up there. So I apologize for that. It was a new section, so I didn't publish the page. I forgot to hit a button. I hate that. But it was definitely up once I noticed it. So I kind of want part of the learning process, and what will really help you guys is if you actually talk about, right, what you're what we're learning about, and you're not always just listening to me. So, does anybody understand bacterial viruses and want to give us a little short, their interpretation or their understanding of how these viruses work? Okay, go ahead.
They're viruses that attack bacteria.
Mm-hmm.
So it like makes that bacteria more powerful. Like if a virus attacks your E. coli, it can like become really deadly. Is that right? No.
No. What happens to us when we get attacked by a virus?
We have to ride it out.
We have to ride it out. But what happens at the cellular level? The virus goes into our cells, and why is it doing that? It injects its DNA so that what can next happen? Right. No viruses, no matter whether it's viruses that infect us or viruses that infect bacteria, none of them could replicate on their own. If you've been in my microbiology class, you should know this, right, y'all? I'm looking at you. Okay. So they've got to get at the very least, their genetic material. In the case of viruses, the viruses that they use, these were specific to bacteria. And they were a specific type of virus that it was DNA. Okay? But we know, right, those of you who have taken microbiology class and other classes, that viruses can also be RNA. But in this case, these viruses are DNA as their genetic material. And in this case, too, just what goes into the cell, which made this an excellent model for proving that DNA is the genetic material. Just the DNA. Okay? So very simply, these viruses, though, are made of just two macromolecules, DNA and, nope, protein, right? So this really complex structure you see in blue, that's all protein. That's basically the delivery device. The red stuff inside, as they have it labeled, is the DNA. So what happens with these particular viruses, right, is that here's the bacteria. The virus, which these guys I think are so cool because they look like little spaceships, huh? They land on the surface and they inject their DNA into the cell. The protein component, right, this capsid part, all the other parts, right, that are made out of protein stay outside of the cell. They do not enter. Just the DNA enters. So this was beneficial because what did the scientists do later with these two pieces? So the protein's on the outside of the cell, right? These capsids are on the outside, the DNA is on the inside. Can we separate these two? Yeah. How did they separate it in their experiment? Something you'd find in your kitchen. You used a blender, right? Basically creating shear forces. So they took these bacteria after they'd been infected by this virus and they stuck them literally in a blender, a kitchen blender, I kid you not, right? And it was in a broth solution, right, a bacterial culture in a broth solution, and they spun it in a blender. And what that did, that shearing force of them spinning around in that blender, ripped these guys off the cell. Then they took that liquid and they stuck it in a tube, and what did they do with it? How would you separate two layers? What machine could we use in the lab? A centrifuge. So again, they spun, but in this case in a way that the cells, which are heavier than those protein components of the virus, would pellet at the bottom of that tube after they centrifuged it. In the supernate, in that liquid phase at the top, would be what? So if the cells are pelleted, what's in the liquid phase? No, the DNA is in the cell, right? The protein coats, right? The capsids all by themselves. Now, one of the problems with this is that are all the viruses they stuck in that test tube going to inject their DNA into every single one of those bacterial cells? No, you're going to end up with some intact viruses which still have their DNA. Which is why when you look at their data, they didn't get 100% separation of DNA from protein, basically. So what normally happens, right, is then the DNA is going to be used as a template to make the protein codes for the new viruses. And the DNA is going to be copied as well by the host machinery, by the host enzymes and ribosomes, right? They're going to make all the pieces. They're going to assemble them, and then these would bust back out of the cells. So, of course, too, they didn't incubate this for a very long period of time because, again, they didn't want a lot of them busting out. They wanted the DNA inside the cells. So how they timed this and how many times they did this is curious to me, but I don't have the time to investigate it. Does that part make sense to you guys? Right? And that's why, too, I wanted you to maybe watch this animation, right? If you don't have the microbiology background or you don't remember from my microbiology class about viruses that affect bacteria, okay? And this is important because not all viruses go to this type of life cycle. So what Aaron was thinking about where some viruses will infect and cause the bacteria to become more virulent, that can happen, right? But this was the type of cycle we were talking about, what's referred to as the lytic cycle, right? And especially for these types of viruses where they actually inject just the DNA. Maybe some enzymes too, but mostly the DNA. And the protein stays on the outside. So that's one part of the level of understanding. Another part is they radioactively labeled this stuff so that they could keep track of it. Because it's really hard to see proteins versus DNA, right? But when we look at them at the molecular level, right, proteins, DNA, most of all of our molecules are made out of carbon, hydrogen, and oxygen. And then additional molecules like nitrogen, phosphorus, and sulfur. And what's awesomely unique about DNA versus proteins is one of those elements I just named is only found in DNA, and the other one is only found in proteins. So, anyone know which one is found in which? So there's sulfur and phosphorus. Who has sulfur, DNA or proteins? Proteins, right? Because 2 amino acids actually out of the 20 that make up proteins, the potential 20, cysteine and methionine contain the element sulfur. So they radioactively labeled the sulfur. And then in another culture, they radioactively labeled the phosphorus. So this tagged what? The DNA. So the DNA, which just has phosphorus, was radioactively labeled. The protein, which just has sulfur, was radioactively labeled. Still with me? So they could use a Geiger counter, right, which detects the level of radiation to determine about how much phosphorus or sulfur was in the sample they were testing. So we went over this already. So phosphorus, specifically in DNA, sulfur in proteins. Right? So they grew them such that they became labeled, so that in one culture they were dealing with, all those viruses, their protein was radioactively labeled. In another culture, all their DNA was radioactively labeled, right? They kept these separate. So only the genetic material of the phage is injected into the bacteria. This is the hypothesis they're trying to prove. So I know it's kind of small, but, you know, sometimes pictures help us. Into the bacteria, right? And so in that, right, we know it's made out of DNA or protein. At that point, they did know this, right? They knew the chemical makeup of these viruses was DNA and protein. The question was, okay, which is it? Is it the protein or is it the DNA that's the genetic material? Okay? So that's why they labeled the protein versus the DNA. So they could tell which one was where. Does that make sense to you guys so far? Okay. It's kind of a cool experiment. Okay. So in this picture, the viruses, it's their protein, right, that's highlighted in red, is radioactive. In this other culture, it's the DNA that's radioactive. So they set up two separate cultures, and they're going to compare them. So they, again, with the labeled viruses, they infect the bacteria just long enough that they inject their DNA into most of the cells. Then they put those cultures in a kitchen blender so that the shearing forces will remove the proteins, right? That protein capsid, little spaceship looking thing that was on the outside of the cell, so it will separate from the cell. It doesn't take a whole lot of force, just putting them in the blender for a few seconds was sufficient. Then they took those cultures and they centrifuged them to pellet the cells, right? And in the liquid phase, what's called the supernate, was hopefully those proteins, those empty virus capsids. So when you look at their data, right, and this is just looking at the supernate, right? So what's in that liquid phase, which we believe, right, is not what was injected into the bacteria, because the bacteria are in that pellet. So in the supernate, there was mostly about 80% of all their samples they sampled. Oh, look, they did different times of agitation in the blender. So 1 minute, 2 minutes, right? So it looks like it took about 2 minutes to get most of the separation, right, in that blender. I wonder what setting, right? Did they pulse or puree or chop, right? What did they use? I'm sure if you found their paper, they probably specified what they did. But so about 80% of what was in that supernate was sulfur residues, radioactively labeled sulfur residues. Right? So for their one that had the sulfur labeled, 80% basically was protein. So that lets us know, as we suspected, right, the protein is outside the cell. They only found about 35% of the phosphorus, and the phosphorus was labeling what? The DNA. And it's probably from viruses that didn't inject their DNA into the bacterial cells, right? Ones that were intact still. So it wasn't 100%, but it was enough evidence, right, that most scientists were like, yeah, you know, this supports all the other people as well that keep saying it's DNA, right? Our genetic information is encoded in DNA. And of course now we have much, much, much more evidence, right? You say DNA, people kind of know what you're talking about. Even general population, non-science majors, right, have a general understanding. So this was a pretty cool experiment, though, right? But it's kind of hard to understand. You got to kind of know some scientific background. You got to understand how these viruses work, understand that proteins and DNA are significantly different as far as the elements go, and that's why this worked. So pretty cool. Definitely a pretty cool experiment, right? So we talked about the 4 levels of complexity. We've basically gotten to the, did we get to the double helix last time? I know we talked about the individual nucleotides and how we put the strands together, right? And what makes up each piece. So a lot of memorization if you haven't dealt with it so far. We didn't get to the double helix, okay. Oh yeah, because we hadn't talked about the other experiment. The one where he figured out A's go with T's and C's with G's, and I can never remember how to say his name. Okay. I need a how you say for all these names. I need to take a class on how to pronounce names. Do they have a class like that? I need that class. Okay, but this is one of the cool things that's in your guys' book. Although I wouldn't necessarily test you like this, you need to know these abbreviations, right? So what is AMP stand for? What is ADP and ATP, right? And it's easier with pictures, right? So we know that why don't they have the letters here? We know the M, D, and P stand for how many phosphates, right? So M is for mono, 1, D is for di, that's 2, and T is for triphosphate. And then the first letter represents the actual nitrogenous base, right? So in this case, A's. So a lot of times we refer to these other two as modified nucleotides. Usually when someone's just talking about a nucleotide, they're talking about just a single phosphate. And that's what you find in DNA and RNA, but sometimes in cells they'll add extra phosphates. And they use this a lot of times as energy currency for the cell. They're storing that energy in those phosphate bonds, and then they're releasing it when they break those bonds. So most of us are aware of ATP and ADP, but you probably didn't actually know what these stood for, right? What the acronym was, right? I doubt anybody actually said adenine triphosphate to you in the past, right? Everyone kept blurting out ATP at you and never actually showed you the molecular structure. So this is what ATP looks like. And so one of the cool things about your book is some of these interactives it has. And so I'm going to show you how to get, am I going to bother? No, I'm not going to do that. I'm actually just going to post this PowerPoint for you guys, I think, that has all the animations in it. Here it is, in PowerPoint form. But you can get to this through your book, too. It's this blank slide. So what you have to do, as it tells you here, is you've got to be in presentation mode to actually even see the slide that I want to show you guys. And then the buttons will actually work. Make sure you turn on your audio. You must click the slide background before you can advance to the next slide, okay? So there are instructions on how to use this. I can't remember if you actually have to be hooked up to the internet or not. I don't know if these are outside links or internal links. But sometimes when you first open it, you might have to enable the content. I probably even did that without even thinking. So that's another thing that'll pop up for you sometimes. So in presentation mode, here's that virus life cycle one. And this is actually one of the Hershey and Chase experiment too, where they walk you through it, which is really cool. They show you the different steps and what they did. Well, part of it anyways. Okay, this is the one I wanted to get to, which was building nucleotides. This is pretty cool, right? So, GTP. So, what does the G tell us? Which nitrogenous base, right? And what does G stand for? Because you guys do need to know this. Guanine, right? Which is one of our purines or pyrimidines. It's a purine. So it has how many rings? 2, right? So it's one of these two guys, right? And it's labeled right here, guanine. You don't have to actually know the actual structure, right? Down to the drawing it out. I don't even know it. I took biochemistry. I still have those flashcards. So that would go right there, right? And then what goes right here that the nitrogenous base is going to attach to? The sugar. So we have 2 choices, ribose or deoxyribose. Which one do you think? Ribose. Because they would denote if it was deoxy by putting a little d in the front. So we know this is a ribose. And it's triphosphate, right? So we have to pick up the 3 phosphates. So then you hit submit, and it tells you, correct, you did it, right? And then as the instructions say, you hit clear to try another one. So this one, notice it has the little d. So which sugar are we picking? Deoxy. And you're right, for the nitrogenous base, we're going to pick cytosine, which is 1 ring, so it's a purine or pyrimidine. Pyrimidine. And how many phosphates? 3. 3 again. And we got it again. So you do have to, obviously I couldn't make you build it, right? But you would have to, like, if I gave you a drawing, be able to know what it is. Or, you know, in a multiple choice question, if I say what makes up DCTP, you would have to be able to pick out that the sugar is the deoxy, right? That the nitrogenous base is cytosine, right? And that you would put 3 phosphates. Does that make sense to you guys? You want to do another one? It's kind of fun. But I'll post this so you can play. There's like, it runs through like 6 different ones and it just kind of randomly repeats, by the way. It doesn't do all the possibilities. So let's clear that one. Let's do one more. So AMP. Adenine, yep. How many phosphates? 1. Which sugar? Ribose. The letter M is for 1, mono. D is di, right, 2, and T is tri. Right. The first capital letter is going to let you know which base, which nitrogenous base, right? And if there's a little d, it'll let you know it's deoxyribose. If there isn't a designation, then it's ribose. Submit. Right, we got it correct. Let's do one more.
Is adenine a purine?
Yep. Adenine and guanine are our purines, right? Because angels are golden, and gold is Ag. One of our tips for remembering it. Works for me anyways. Okay. So, all right. What do I need to put? Okay, thymine in the base spot. Deoxy because of the little d, right? And how many phosphates? 3 because it's a T, right? Triphosphate. Submit. We got it right. All right, one last one. Uracil. Ribose. Triphosphate. Submit. We got it right. Clear. Right, and you can keep going. I have a lot of T's in this one. This is another one that GTP, as used by our cells, is energy carrier other than ATP. But again, the only difference we know now is what? What's the difference between ATP and GTP? Guanine and adenine, the nitrogenous base. It's the only difference. That's it. Okay, so this is kind of fun. I'll post this PowerPoint for you guys. And then it has a cool animation on Watson and Crick's thing. Actually, this one is pretty cool. I might just play this one right now. Let me plug it in. Because this is our next section anyways. And then hopefully they'll say that name for me so I know how to pronounce it. Alright, so let's see. Just got to make sure I have my volume on in the volumes there. And it seemed kind of loud, so it might blare us. Let's see.
The structure of the DNA molecule was first inferred by James Watson and Francis Crick, based primarily on X-ray crystallography data collected by Maurice Wilkins and Rosalind Franklin, and chemical analysis of base composition of DNA conducted by Irwin Chargaff.
Chargaff.
The key features of the structure are its right-handed double helical structure. Each helix consists of an alternating sugar phosphate backbone with nitrogen bases projecting toward the interior of each helix. One complete 360-degree turn of the helix covers 10 bases in length and equals 3.4 nanometers in physical distance along the axis of the molecule. The width of the double helix is 2 nanometers. The nucleotide bases are attached inside each backbone of the molecule so that the nucleotides in one helix or strand are hydrogen bonded to the bases in the other helix or strand. The hydrogen bonds hold the 2 strands of the double helix together. An adenine in one strand is always paired with a thymine in the other strand, and a guanine in one strand is always paired with a cytosine in the other strand. Note that guanine-cytosine base pairs form 3 hydrogen bonds, while adenine-thymine base pairs form 2 hydrogen bonds. This makes guanine cytosine base pairs more stable than adenine thymine base pairs. Nucleotide pairing between strands also allows the sequence in one strand to determine the sequence in the complementary strand. The two ends of the strand are not identical. Note that on one end of each strand, the 3' hydroxyl group of the deoxyribose sugar is not involved in the backbone, or we say it's free. While at the other end of the same strand, the 5' hydroxyl group of the deoxyribose sugar at the end is free or may contain a phosphate that is free and not bonded to another deoxyribose sugar. This dissimilarity of the two ends of the strand creates the ability to uniquely distinguish each end of the strand. Because of this polarity of each strand, we can see that the two strands of DNA are oriented in opposite directions, or we say that they are antiparallel. In three-dimensional space, the two helices are not equally spaced along the axis of the molecule, such that a major groove and a minor groove are created by the asymmetric spacing of the antiparallel helix backbones.
Wow.
The major groove and minor groove play important roles in the binding of proteins that regulate gene transcription.
So proteins actually will bind in those grooves. And when you look at the DNA, especially when you look at a longer strand, you can really see. Come on, mouse. Can't even find my mouse. They said I had to click off of it. I couldn't even click, period. So when you look at a longer strand like you see here, do you see how there's like a bigger gap here? Right? And big gap here, and then not as big of a gap here. So the big gap is, of course, what? The major groove. And the smaller gap is the minor. And it still isn't even a long enough piece to see it. We'll see if in my PowerPoint if we got a better picture. One of the things that I was hoping they would talk about in this, and they don't, is they've got a picture though. I know I saw a picture I'm looking for. At least I thought I did. There it is, there it is. Okay. So, do you see these kind of like blocks here? Do anyone know what they're representing? What part of the nucleotide? Yes, the nitrogenous bases. Okay? And there, if you were to look at this, this is why some people will sometimes describe this as a spiral staircase, is these almost look like the way the bases orient themselves is so they're kind of flat like this, if you were looking at it straight on like we are. They almost look like steps, don't they? Right? So imagine that like those are the steps to your spiral staircase, and then along the edge here, right, are like the railing and the handrail, right, the support of a spiral staircase. Can y'all see that in your mind? So those are made out of what part of the nucleotide? What's here on the outside? So if these are the nitrogenous bases, what's all of this making up the ribbons here? The phosphates and the sugars bonding with each other, right? And remember, what is that bond between the phosphate and the sugar called? So there are phosphates involved, and then another organic chemistry term. Phosphodiester, right, is the name of that bond. If you're in genetics class, yeah, you got to know a little biochemistry. Okay, especially molecular component of genetics class. Okay, so a little bit. You can handle it. I'm not asking you to explain the bond. Just remember the name of the bond. Okay. All right, so when we look at this, right, so we see those different grooves, the major and the minor. We'll look at that a little bit more. So the other thing they talked about is that if you were to follow this up 360 degrees, right, one complete turn, that distance would be 3.4 nanometers. Because all of these things, right, are pretty uniform. Right? All the sugars are the same size, the phosphates are the same size, right? So you end up with this equal spacing. Does that make sense to you guys? between, say, the steps. So if you were to count the bases, there would be 10 pairs, right? 1, 2, 3, 4, 5, 6, 7, 8, 9, 10 pairs. Right? 10 steps of that spiral staircase. Still with me? Okay. So then each strand, they're connected to each other by the base pairing. And we know, and this is how Watson and Crick helped figure it out, because of this guy, Chargaff? How do you say that? Say it.
Chemical analysis of base composition of DNA conducted by Irwin Chargaff.
Chargaff.
The key features of the structure.
Chargaff, right? So he did chemical analysis where he took, and we'll look at that chart in a moment, he took a whole bunch of DNA samples from a whole bunch of different organisms. They just give us a little snippet in our book. And he separated out the A's, the G's, the T's, and the C's, and figured out about how many there were per the DNA sample. And so what he found out from that is the percentages of the G's and the C's matched each other pretty darn closely, and the A's and the T's. So that's how they were able to surmise, that's how Watson and Crick were able to surmise that A binds with T and G with C. And of course they figured out from Rosalind's work, right, in Maurice's lab, that from her X-ray chromatography that it was a double strand, right? So that we had this base pairing going on. Still with me? The other thing we now know is that when these two bind with each other, the A's with the T's and the G's with the C's, the guanine and the cytosine and the adenine and the thymine, is that between the A's and the T's, they form 2 hydrogen bonds. Between the G's and the C's, the guanine and the cytosine, there's actually 3 hydrogen bonds. So depending on the sequence, if you have more G's and C's, it's said to be, some scientists would say, GC-rich code, right? If you had more G's and C's versus A's and T's. And there are actually some sequences where specifically there are more A's and T's, like the Tata box you may have heard of, right? This is a specific segment in the DNA where there's a lot of repeats of A's and T's. And the reason for this is a common section of the DNA where the 2 sections separate from each other. Because think about this. Breaking 2 hydrogen bonds is much easier than breaking 3, right? So if you have a lot of A's and T's, that's not a really strong bond, right? If you have a lot of G's and C's, that's going to be much stronger bonding, much harder to separate those 2 strands. Still with me? So as I said, some scientists will actually refer to sequences of having a lot of GCs versus ATs, right? If it's GC rich, there's a lot of G's and C's. Well, especially for molecular scientists like myself, when you're trying to separate those 2 strands, and if there's a lot of G's and C's, you're going to need a little bit more energy, right, to separate those 2 strands. So the other thing that they go over here as well, and we've already talked about, is this bonding, this phosphodiester bond between the sugar and the phosphate. But I'm going to zoom in on that a little bit later. But one of the things I want you guys to notice is, as I said, the orientation, right? So it almost like these guys are right side up and these guys are upside down, right? They're anti-parallel. They go in opposite directions from each other. And that's important because the enzymes that we use are going to bind to these molecules based on orientation. So ours read in what's referred to as the 5' to 3' direction. And remember, 5' is that carbon on the sugar here that doesn't have another sugar phosphate bond to it. And this one's known as 3' because the 3' in that 3rd carbon here also doesn't have another molecule binding to it. It's free. Where notice flipped over here, right? It's 3' to 5'. So this is why we have the leading and the lagging strand, right? When our DNA strands separate from each other and they're being copied, the leading strand, the enzyme can just run right down. It just runs right down 5' to 3'. Where the other one has to, as it's coming, the DNA is being separated, has to keep backtracking basically, right? Because he goes in this direction, right? 5' to 3' is this direction. So once these are separated, then he goes in here and he fills in this direction. Then once it's separated a little bit more, he goes down to the beginning of the separation, right? And then you have the Osaki fragments and everything like that, right? But that's the reason for that. Why that happens like that is these enzymes read in a directionality, and the direction of these two strands is opposite from each other. Why that is, I have no earthly idea. I don't know how to explain that, but it is what it is. Okay, what else did I want to point out here? Yeah, it doesn't do a good job with the minor remainder. Okay, so I'll post this presentation just how it is for you guys, so you can download it and watch it and play with it. I'll put it on our site. It's really handy to have it in that format. So there's our phosphodiesters, 5' to 3'. So the key experiments with the double helix, so the last one that we hadn't gotten to yet was Irwin. I'll refer to him by his first name. I can't say his last name. Chargaff. And so, as I said, he separated out the A's, the G's, the T's, and the C's by chemical analysis. He was a pretty cool chemist, I guess you could say. So here's some of his data. So when you look at this, as I said, this is just a sampling of it. You can see, as we know, the percentages of the A's and T's pretty well match each other, and the G's and the C's, right, pretty well match each other. Now this is, actual data, so the numbers aren't perfect. But, this data was available to Watson and Crick, and so this really helped them in figuring out the structure of the double helix, along with the information from Maurice and Rosalind. And then, of course, I don't think we talked about the other guy, Linus Pauling. He was studying and making ball and stick models with proteins. And so what he was doing with proteins, they used that information to help them make their ball and stick structure of the double helix for the DNA. So it really wasn't, they used, like scientists do every day all the time, right? The only problem people have is, the fact that Rosalind didn't get credit, right, for them using, her work, and how instrumental her work really was for them solving this puzzle. And that's really what it was. It was a puzzle, right? They were trying to figure out how the pieces went together. They knew all these pieces, right? But how do we fit it together? And they figured that out. So this is also sometimes referred to as the Chargaff rule, right? This percentage of A's to T's and G's to C's. Those percentages are going to match up. So in 1962, right, they were awarded the Nobel Prize. Watson and Crick, get most of the credit, but Maurice was also on that award. Sadly, Rosalind wasn't, because she had already died. So this is some of the stuff that we saw in that animation that's important as it relates to the structure of the double helix, right? You can imagine that there are like 10 base steps in that one complete turn, and it would measure 3.4 nanometers. The two strands are what we call anti-parallel, right? So there's opposites in directionality there. The helix itself, the way it turns, is normally right-handed or clockwise, right? So you go to the right or clockwise. I don't know, I'm dyslexic, so dealing with the clock makes more sense to me. But that's right-handed. So as it spirals away from you, if you were looking at it, so like if you were looking down a spiral staircase, it would go clockwise. Can you all envision? Okay. or what's referred to as right-handed. Normally. So we know the complementary base pairing, right? And again, I tell myself little stories to remember these. I always remember AT&T, which I'm surprised that company is still around, but they are. They've been around for eons, right? AT&T. So I just remember A's go with T's, like AT&T. And G's and C's kind of look like each other. They're both curved letters, so it just kind of makes sense that they go together. So that's what's worked for me. Just really that simple is how I remembered A's goes with T's and G's with C's. Some people have told themselves little sayings. Some people know little memorized sayings. I'm not that complicated for these guys. But whatever works, right? Whatever works. So base stacking within the DNA, the bases are oriented, and that's what I was really trying to show you in that one picture where they look like blocks, that they would be flattened. You know, you wouldn't be seeing their rings, you would be seeing them from the edge if you were looking at it like a spiral staircase. So literally just imagine the steps within the staircase.
They're both bonded by hydrogen bonds?
Yes, but the difference is between the G's and the C's, 3 hydrogen bonds, A's and T's only 2. So you have the 2 grooves, and yeah, again, you know, you can see how there's like the bigger spacing here as opposed to here between the strands. So yeah, this picture just doesn't, I don't know. This picture's not making me happy. But you can definitely see how it's a much bigger gap there. Right, in this picture, that's the major groove, and here's your minor groove. Not as much space. So it's not perfectly symmetrical like a spiral staircase would be. Does that make sense to you guys? It's still beautiful, though. All right, so for the most part, that double helix, what we call a secondary structure, that helix, the predominant form is what we refer to as B DNA. Why B as opposed to maybe A? I don't know. There's actually A DNA. And then there's also, under certain conditions, Z DNA. And this one got its name, when you look at the picture, it makes kind of sense why it got its name. But the difference here is it's not right-handed turning, it's left-handed, so counterclockwise, if you're to look down the spiral. And its formation is favored by having really rich GC regions at high salt concentrations, and where the cysteine is become methylated at low salt concentrations. And you have what's referred to as negative supercoiling. And this is where I think the best way to visualize supercoiling, especially for circular DNA, is if you took a rubber band and then twisted it upon itself, you get that extra layer of twisting. But imagine that that rubber band itself was a double helix. So you've got twisting on top of twisting. So they believe it may play a role in transcription and chromosome structure. So here's the picture of it. So here's B DNA, right? That normal, we're used to seeing the major groove and the minor grooves. This one's turned the other way, spins in the other direction. And so because of that, that backbone on the outside here, it tends to have this zigzag pattern. And so that's where it gets the name Z DNA, because of the zigzag. And it's more condensed. Do you see that? How it's tighter packed than the B DNA. And the bases here in the normal form, the B DNA, they're perpendicular to the central axis. Like if you were to put a pole right up the middle, right? So for like a central axis to a spiral staircase, they're parallel, right? They're the steps of that staircase. Where notice in this one that they're tilted. See how they're tilted? Some in this section are tilted this way, others are tilted that way. That would be like a funhouse set of spiral staircase, right? And the only reason that would exist is because of mirrors, not because it actually would be tilted like that. It wouldn't be structurally sound to be truly tilted. They'd have to be, modified in some way. So, I feel like I forgot something. Nope. Okay. So the last thing about DNA, as far as the helix goes, is that they've actually found that a triple helix can form. So where you have the double helix and another additional strand of DNA binding in those groups. So this was found in the 1980s, and it was using synthetic strands. So whether this actually happens in nature or not, it may or may not happen. But we have some evidence to this, because remember we talked about the bar bodies and how RNA would actually bind to the DNA in those regions and kind of compress it. So as you can see here, here's the double helix, but here's an additional strand, right, that's binding within the grooves. So it's represented here by the purple. You see that? It's kind of cool. And again, it's base pairing. How, it's kind of a mystery. So we save the best for last, right? RNA is the best. Some people might say that. I didn't like it so much in research. There are naturally occurring RNases in the environment that destroy RNA. So when you're a molecular scientist like myself and you're working with RNA, you have to be super, super clean. Like insanely clean. No, not one of those suits, but all your stuff needs to be kept clean, and like you have to buy special pipette tips that have been treated so they don't have any RNases on them. So if someone else comes in the lab and messes with your stuff, they could screw up your whole experiment by just touching your stuff with their hands, because your hands has RNases on it. So I would always have to wear gloves, and I'd have to work in a little clean hood with all clean stuff. Otherwise, my RNA would just get chopped up by RNases.
You worked in a hospital?
No, I worked in a research lab. I did research for my master's degree at Southeastern, and my research was in RNA. So we were looking at the expression of RNAs for some genes involved in the immune response, known as the RAG genes, and we were looking at the effects pollution and bioterpene had on the tadpoles that we exposed to those chemicals, how it affected their expression. So I didn't actually finish the project though. My primary professor went a little cuckoo and I bailed ship. So, and she hasn't had anybody graduate since I left. So, yeah. Not finishing a project. She's only had one person in like 10 years finish a project. She's a little crazy. So I finished a different project and got the heck out of there. Which didn't get published because I had to have her permission, and I don't want to talk to her. which is really bad as a scientist, right? I should, I would have 3 sequences in GenBank because I did sequence the DNA from 3 different frogs for these genes.
Do you
But
believe that the population has like genetically mutate cells? It's like they say in Baton Rouge, around Egyptian, that they call it Cancer Valley. And a lot of people around there have.
there's a lot of scientific evidence to the fact that chemicals mutate DNA and therefore can lead to cancer. Absolutely. Absolutely. Lots of evidence. So what are some key features of RNA that you guys already know? So what do we know as compared to DNA? What's the difference?
Sugar.
The sugar, right? The sugar is ribose instead of deoxyribose. It's not missing that oxygen that ribose typically has, like the sugar in DNA. What else, Tyler?
It doesn't have thymine and has uracil.
Right, doesn't have that nitrogenous base. It doesn't have thymine. It has uracil instead, but it does have adenine, guanine, and cytosine. Excellent. One more thing. We've been talking about structure a lot, right?
Say like the result of it is a different form. Because like DNA kind of results into RNA, which is a form of nucleic acid, whereas RNA is going to result in protein.
Right, okay. So there is how it's utilized by the cell, right? DNA is storing the genetic information where RNA is more transfer of that genetic information, right? Somewhat, yep. So its functionality is different. We're still on structural though. Structurally, how is it different? It's single-stranded, right? It's not double-stranded, it's single-stranded. It can interstrand bind, but again, it's not more than one strand. It's just a single strand, right? So we'll look at a picture of that. So I think you guys hit all the major ones. The primary structure of RNA strand is much like DNA, right? But the strands are several hundred to several thousand nucleotides in length, where we're talking about millions, right? Or a couple thousands, or much longer for DNA. In RNA synthesis, only one strand of the DNA is used, where when we copy DNA, both strands are used, right, as a template for making the new DNA. For RNA, we just use one strand of the DNA as a template. So structurally, the sugar, as we said, is not missing that oxygen on the 2' carbon. It is a complete ribose sugar. And instead of having thymine, we have uracil. So what is the complementary base pair to uracil? Adenine, right? And then we still have G's with C's. So there's just a switch out there. Instead of a T, we're putting a U. We still have that phosphodiester bond, right, between the sugar and the phosphates. It's still 5' to 3', right? It still has directionality, except we only have one strand. Although it's usually single-stranded, it can form some short double-strand regions, but this is due to complementary base pairing. And we'll look at a picture of this. So it's not actually 2 strands, right? But it's the one strand binding with itself and forming a double section. I don't know how else to explain that. It's right-handed, right? So it still kind of curls, helixes, like DNA does. It's just not 2 strands. Okay, it's not a double helix, it's a single. Sometimes. And in that case, it's 11 to 12 base pairs in each term. So it's a little bit longer. It's in the A form. So maybe that's why DNA is B. Maybe they figured out RNA first. I don't know. So they have different secondary structures other than the helixes they can form. They can interstrand base pair with each other where they're complementary, and sections that aren't complementary tend to bulge out, right? And so we refer to these as loops, and this one really bulges out, right? So it's called a bulge loop. or where this one is more of an internal loop. And this is a multi-branch junction, almost like a four-way intersection. And this one, there's a section where they're base pairing off of the main section, but then you still have that loop. So this one's called a stem, because it looks like it has a stem and a loop. And this structure is very common in a particular form of RNA known as transfer RNAs. So for transfer RNAs, you have some of these sections, right, where you get this double helix where the two strands are interacting, two sections of the same strand are interacting. I don't like that they call it a double. It's double as in it's two strands, but remember that the RNA itself, if you just stretch it out, it's just a single strand. Does that make sense to you guys? Right. But then we could take part of those pieces and they can interact with each other, two strands, and they can form double helixes. So this is what's called a ribbon diagram. But what's important is that there's a portion here somewhere, I guess somewhere in the middle of the strand, you could say, that's referred to as the anticodon. This is opposite, anti meaning opposite, of the code found in the messenger RNA that depicts on which amino acid is going to be placed by the ribosome. So the transfer RNAs do what? They actually transfer in the amino acids. So for each transfer RNA, its codon sequence determines which amino acid it would have here at this other end of the molecule. So as these guys transfer in, this portion matches up with the messenger RNA, right? So it matches, right? The ribosome's like, yep, this is the right one. And then the ribosome takes from this end the amino acid and hooks it to the amino acid before it. And then it keeps adding on to that chain of amino acids as it builds the protein. So this is really important, right, for transcription, these transfer RNAs, because they actually transfer in to that complex of the messenger RNA with the ribosome the correct amino acid based on the code that's in the messenger RNA. And where did that come from? The DNA, right? And that's why it's called messenger RNA, because for us, especially for eukaryotic cells, it travels from the nucleus, where the DNA is, to the cytoplasm or the endoplasmic reticulum, the rough ER, that has the ribosomes attached to it, or ribosomes in the cytoplasm. Those bind to the messenger RNA, and they're going to read that sequence. It's actually the transfer RNAs that actually transfer in those amino acids that match up with the code. And that code in messenger RNA is read in groups of 3 nucleotides known as a codon. So that's what this anticodon is. It's just 3 nucleotides. That's it. So this particular one carries the amino acid phenylalanine. So when they abbreviate it, they write tRNA, and the PHE is a 3-letter abbreviation for the amino acid phenylalanine. That's how we shorthand for a specific RNA. Transfer RNA, that is. So there's my little saying, right? Angels are pure, like purines, and they have 2 wings, like purines have 2 rings. And angels are golden, right? Adenine and guanine, Ag, which stands for gold on the periodic table. So, but again, you can tell yourself whatever stories you need to remember the information, right? For this course. So that's it for Chapter 9. It's a lot of stuff, but hopefully some stuff that you guys know previously and maybe understand better now than before. So I had already put together the Connect assignment. I'm just going to post it for you guys for this so that you guys can, when you feel comfortable, practice your knowledge. You'll notice on our list, we're actually only covering one section in the next chapter. It's section 3, I want to say. And so I'll have that posted for you guys later today, the PowerPoint for that. don't want to be wrong. But, yep, 10.3. Eukaryotic chromosomes.